Welcome to the Axial Podcast. Axial is an early-stage investment firm based in San Francisco. We partner with great founders and inventors investing in early-stage life science companies often when they are no more than an idea. Axial is fanatical about helping the rare inventor who is compelled to build their own enduring business.
Okay, I think we're recording. It's always really weird to press record, isn't it, Justin? So we're really excited to have you. Have you on this podcast. I think you're number 4 or no, number 8 actually. I did a bunch at the beginning, but yeah, really excited to have you. You're one of the geniuses of biotech, but you haven't had any social media presence ever. So this is kind of the beginning of a new era where people finally realize how smart and talented you are, and get more visibility for Serotiny. So hope you're doing well. How are you?
I'm doing good. Be fun to chat. It's always a little strange to make things public, but I enjoy it, and I enjoy chatting with people. I enjoy the community. So see what we can do.
Exactly, Justin Farlow. Justin and Colin Farlow. You guys like the Wright brothers, but for proteins. So you're gonna make proteins.
Can't forget Colin.
You're gonna make protein slides. Everybody loves brothers in America. It's American. It's such American. 2 brothers building companies. It's just part of Americana. So you got to tell your story. You and Colin, you're gonna start taking like Patrick John Collison pictures one day. Yeah, it's gonna be dope.
It'll be good. I hope so. We tend not to take the fancy pictures, just like the social media, but we have the good time. So we enjoy it.
You're gonna do it you're gonna do it. This is the beginning of a new era for Justin Farlow. Seriously, this is it. This is the round whatever. You're coming. This is like you're coming out. This Diana Ross song. You're coming out. Or I'm coming out.
Oh man.
This is you. But let's talk about. Okay, we're gonna talk about your background first, and we're gonna go into Serotiny, and then we're going to kind of a lot of your great ideas are in SynBio, and we could probably talk for a few hours, but we're gonna keep it short. So we're pretty busy. So yeah, to start with the conversation, we'd love to I think for everyone's sake, especially the up and coming scientist and founder, kind of your background. How did you get into science, biology? How did you learn? One thing about Justin, not only an incredible immunologist, biologist, but like a world-class programmer. How did you do both of those things? So whatever you want to talk about, I would love to hear your kind of origin stories.
Part of science itself, more just a curiosity of how things worked, philosophical curiosity. So I actually it wasn't obvious to me that I was going to be a scientist early on, but I really enjoyed trying to understand how things worked. So a lot of the early work I did studies I did were in physics and in chemistry, trying to really understand that. And biology only came later. I actually studied a lot of the philosophy of science, and I found that to be almost as interesting as the science itself, in different tools and ways to get at what was important or what was foundational. By the time I got to grad school at UCSF, the biology had, I think, reached a point where you could start to program it, you could start to think about it in a technical way as an engineering discipline, as opposed to more of just a cataloging discipline. And that really intrigued me. So I had a little bit of background, had a bunch of friends who were really good at programming, even in middle school and high school, and they taught me how to program. And with that, built all sorts of little utilities and fun little things, but never professionally. But that was enough to be able to be part of the community. And I found these communities helpful. And being in San Francisco is part of that community of programmers as well as scientists. And then things like Hacker News and Y Combinator, I think, have done a really good job of allowing people to think about the technology and software engineering and programming in a way that really inspires people. So
cool. Yeah, I mean, Justin's a really active participant of Hacker News. So if you check him out
more than chat, but that's
check his comments out. I think he's pretty good. He has good comments. He's very thoughtful comments. And I remember talking to Colin, and he was like, "So Justin just picked up like Objective-C real quick, or things like that, or he knew how to just code in Golang." I think Serotini's coded in Go still. I think.
We do quite a bit in Go. It's a fun language to use, but best tool for the best job. So.
Exactly. Okay, and so you guys grew up in Indiana. I love people from the Midwest. I grew up in Orange County, but something about the Midwest is romantic, actually, to me. I don't know why. It's just growing up in a boring area with open fields, right? So it's a great place to grow up because it's safe and you can do anything you want. And so, what was the kind of going from Indiana to San Francisco? Was there a logic there to pick UCSF? Was it a specific lab? Was it part of a grand plan to start a company, or kind of what was the logic to go from Indiana to SF? And this is what, like 10 years ago, I think. So
maybe a little more than that.
More than that, actually.
I found Indiana. What was nice about it is it was full of life, literally. Again, I spent a lot of time outside in the woods, surrounded by plants and animals. I didn't find that boring at all, actually. I really enjoyed that. At UCSF, it offered for grad school. So I guess to step back and to be clear, Colin's my brother. He helped me co-found Serotiny, and we both grew up in Indiana. Both went to Indiana University at Bloomington. And while I was there, Bruce Alberts, who was one of a few prominent professors at UCSF, had helped create a program called the Tetrad program. And he was actually giving a talk at UCSF. It was not actually for the students, it was actually for the faculty. And it was about this idea that many grad students didn't necessarily want to continue in academia, and that was hard for the professors themselves to acknowledge. But it was an important acknowledgement, and when you could acknowledge that, you could actually help the students even better, but you could also help society. And so the Tetrad program at UCSF had deliberately carved out space to make it useful for the grad students to teach or to think about public policy, as well as business or law. And so they recognized that not every grad student would become an academic. And I appreciated that. At the time, I actually thought I would be a more academic scientist, but I really appreciated that idea that scientists should be part of the greater community. And I actually didn't know about UCSF until he was there. I'm like, but that's the program I wanted to think about. So
cool. Bruce Alberts, he's a legend too. Bruce Alberts, everybody, he's like the biochemistry textbook king or something.
Absolutely, truly on the textbooks, literally.
He's on the textbook.
And a really good guy too.
Yeah, he's pretty nice. I've never met him, but I saw him on, like, I see his lectures. He seems like a pretty chill person. So you're UCSF. Mm-hmm. And I might get some shit for the Indiana thing. I love the Midwest. It is kind of boring, but it's beautiful. It's beautiful though. I might get some crap for that, but so well. And so UCSF, you're working in Zev Gartner's lab. Zev was like the first grad student for David Liu's lab and Bertozzi's lab. Mm-hmm. What was the kind of logic to join Zev's lab at the time, and kind of maybe touch upon your research there and how that?
Yeah, I rotated through a number of really wonderful labs. Zev was a young professor, and I was one of his first grad students, and it offered a freedom of projects, and in retrospect, a diversity of projects, because I helped to work with a number of my colleagues, and that was a wonderful opportunity to not have just a single project that I called my own, but to be able to have inputs and to learn from my friends and peers. So the projects were interesting. I didn't realize at the time quite how much of a synthetic biology approach that we were going to use in that work. But I really enjoyed that.
Cool. Yeah, Zev, it's always good to join a young lab, and you're maybe talk a little bit timing then. Maybe it's one thing about your scientific career and that set up Serotiny is you joined UCSF at a really interesting time. This is when CAR-T was coming online. This is where, Wendell Lim's work, a colleague of, Zev Gartner's work, kind of started Cell Design Labs, got acquired by Gilead, and you've seen a lot of success with CAR-T. Can you talk about timing?
Yeah. So that actually happened later. So at first, it was really fun to see the engineering aspects and to see what were kind of considered toys, I guess, actually working. So I remember one of the fellow grad students who was ahead of me, like Anselm Levskaya, had helped to create some of these optogenetic sensors, and it was just incredible that you could take a protein from a plant and put it in E. coli or in yeast, and now the yeast or plant would respond to light in ways that you could program. You could tell the new organism or this organism how to respond genetically in different ways. And that was really interesting to me because that started to look like the programming that I was used to, and it kind of spurred that engineering mindset. And it turned out this was happening not just at that point, but there were other features being built, new toys that were starting to be programmed. And this was the beginnings of synthetic biology. And it had been going on for a while, but I was watching it, in my neighbors. And about halfway through, Cas9 was discovered, and we saw lectures about why that could be interesting and could be the future before it was. And same with some of these other synthetic biology tools that actually became useful and actually became what could be therapeutically useful. So it's fun to watch that happen.
Yeah, I think this is around the kind of 2012, 2014 timeframe, where kind of a lot of these new modalities are having some success or getting discovered, and so you kind of seeing this, you're kind of developing a set of tools that are related. Now we've established, Justin's a genius, multidisciplinary, curious, studying philosophy and biology and everything. One thing about Justin, he can talk about anything. I can talk to Justin about anything, actually. We talk about Chinese history and the Warring States, maybe, and we also talk about physics of something else. So that's one thing about Justin. He's really fun to talk to. Now we established that. Let's talk about Serotiny. Let's spend the most of the time talking about Serotiny. Kind of, you're at UCSF, you're building these sets of tools, engineering various proteins. You're seeing colleagues have success in, either CAR T cell therapies or gene editing. You've seen some early signs there. What was the motivation to start Serotiny? How did you get the guts to start a company? And yeah.
I think one thing was watching others do it, and I know this is becoming a theme, but when you see that it can be done, you're like, oh, I could try that. Internally at UCSF, it's the same crew of people who had that insight, but Reg Kelly, who I actually met right as I was interviewing at UCSF, had gone on to create what became QB3, and the ability to incubate companies out of the UC system. And Reg Kelly did part of it, and Doug Crawford did the other. And so they had a program to create a startup in a box. They'd help you incorporate, help you get set up, and they'd really help you start to get those first grants from SBIR grants. And at the exact same time that I was graduating, my brother, Colin, was graduating from law school in Madison, Wisconsin. And not only were there companies coming out of the bio side of things, but in San Francisco, you're also in kind of the newer version of Silicon Valley. You're a little further north than the true Silicon Valley, but you're also surrounded by companies. And I could look out my lab window and I could see, Reddit's headquarters and Adobe's over there, and Uber's over there and Lyft's there. It's like, okay, well, just try it. So Colin and I initially created Serotiny as a software company to make the design of those synthetic tools that I really enjoyed, those synthetic biology proteins, the tools to make that efficient, the design of those objects efficient, the design of those objects predictable, and to couple the design with what you actually wanted to accomplish. A lot of the design tools at that time were, how do you manufacture it? How do you build the DNA and make sure the DNA works? But a lot of the end product that you were working with was the protein that was encoded by the DNA. So you want to know the properties of what you were at the end of the product, rather than the properties of the manufacturing. So that's really, we built that software to start off with.
Cool. Yeah, you definitely were living the full San Francisco experience. I think you guys. I remember you living in Tenderloin for some time.
Oh yeah, on a couch and rent. Definitely the full San Francisco experience.
Full Justin and Colin have the full
outrageous
SF
rent
experience. Paying really high rent in the Tenderloin. Now Justin, you live in a really good neighborhood now. I'm really happy for you. You're safe now. I remember you guys living in Tenderloin. I was a little nervous for you. I was like, oh yeah. But cool. Let's talk about Pinecone. I think you were calling the software Pinecone at the time, and maybe you still are. And so, how did you think about defining the problem set? So you kind of have this inspiration to start a company. You're seeing kind of opportunities in protein engineering in general. For Pinecone, how did you define a problem set, and how did you iterate across building what people want? Because at the time you had other type of tools out there. How did you end up finding applications for kind of this generalizable software you were building out?
It was initially motivated by my work as a grad student. I happened to be working on a protein called Notch. Notch is a ridiculous protein. It is a transmembrane protein. It is enormous. It is floppy. It is cleaved. It has every problem that any protein engineer would not want to have in the protein that they were studying. I didn't quite realize that until later, but it forced me to have to be a little creative and made a lot of tools that other kind of protein engineering efforts might have. Those tools weren't accessible to me. But what was also really interesting about Notch at the same time was Notch was a philosophically interesting protein because it was made of components, and the components are found in single-celled organisms. But when you put those components together into one protein, Notch is likely the protein which allowed for multicellularity to come about. So philosophically, it has all the components that all other organisms have, but if you rearrange them into the right way, you're actually able to get this enormous explosion of function of life itself. And so taking a little bit of inspiration from that and saying, what if we could decompose or deconstruct the proteins, both of Notch, but these other proteins, as well as from the rest of the proteome, could we rearrange them and create new proteins that gave human cells new functions? And again, I was watching that happen over and over again, watching simple things like GFPs and tags and halo tag and clip tag be used to these effects. And it was very hard to say, what's the best one? Where should I put it? How should I do these things? And these kinds of protein design had nothing to do with a structure of a protein. It was very much trying to figure out how do I build and make the cell have the function that I want.
Cool.
So we built the software to catalog the components, to make the components easy to understand. GFP, rather than having this long, enormous sequence that is not readable by a human, we gave it a little blob that was green, because GFP is green fluorescent protein, so it should be green. And we didn't know, or we don't know as humans, what many of these proteins look like, because these are all the proteins that don't have crystal structures. So we would just give them icons, and we'd give them unique icons, and that gave it a game-like creativity where you could start to, when you're a grad student, you build your little presentations and your posters, you empathize with the creatures that you've made, even if they're little proteins, and you spend 6 years playing with these little creatures. And if you can make them empathetic and you can enjoy them and you get attached to them. And I felt that, one big circle representing your protein that you spent 6 years on didn't give you the kind of empathy you needed for something you spent so much effort thinking about. So we tried to give all the little components some kind of feature that we could work with, and that helped us build and design and catalog them.
Okay, cool. Because this context, Notch is a very important developmental pathway you learn, and Bruce Albert will teach you that or something, I don't know. And Notch is a receptor, and a delta is the ligand, and it leads to a lot of signaling, and then you can create synthetic versions that lead to different orthogonal signaling pathways in cells. And think how SynNotch, that was really big at UCSF at Wendell's LIMS lab, and that was kind of useful for various applications. And just move into then kind of this idea of designing multi-domain proteins. You're alluding to, right? These big proteins have multiple domains, really large, hard to crystallize, and if you want to create synthetic versions, you create a whole different catalog of different proteins that then are almost impossible to crystallize at scale. And so you kind of need a new language to analyze these kind of large, complicated proteins that have a lot of high value. And you kind of talking about this too in terms of labeling and creating creatures. Okay, that's cool. That's a cool analogy. How did you, when you were starting Pinecone off and talking to customers and thinking about the problem to solve, for especially this big problem of designing a multi-domain protein? Ultimately, that's what Serotiny is doing, right? You guys design multi-domain proteins using machine-guided evolution. That's your term. It's a good term, right? I love it. And that's so, right. how did you think about initially that common language for designing these type of proteins?
Well, what was happening was Colin and I, we'd sit on the couch and we'd be working on the software, but the data that goes into the software are those proteins, they are those machines. And every time a new paper would come out, we would go and try to find the sequence, which was stupidly hard sometimes. But once we found the sequences, you realize this fancy new paper that gave a fancy new name to something is actually just recombining 3 objects, putting those 3 together. It's like, okay, cool. Put that into the software. And these just kept coming out. Every week you'd have new tools, new objects, new proteins made in this way. It became clear that there were a few that were financially or therapeutically useful. So there were a lot of proteins that were being made for basic discovery, and we actually did a lot of work there, and we had some really great collaborations. But as a business, it became clear that there were some of these where if you could create new ones, you could actually help out therapeutically. Specifically, the 2 real big categories were the gene editing tools, like the base editors and the prime editors, all those things attached to Cas9, and then the chimeric antigen receptors, the CARs, and for going after cancer by changing the way a cell behaves. And in both cases, you could simplify the fancy new tool that undergirds billion-dollar companies to be a set of protein domains that have different functions. Cas9, the piece of CRISPR that allows us to edit a human genome, has 2 main functions. It can seek and find a specific DNA sequence, and then it can cut there. Turns out the seek and find is actually a little more difficult and a little more interesting, and you can change it real quickly just by making a new RNA. The cutting actually is not so desirable. You don't really want to cut DNA. So then it became logically the next step is to figure out, well, what do you want to do when you get to a particular sequence that you like? You don't want to cut. Maybe you want to change, or maybe you want to recombine, or maybe you want to integrate. And so each of those ideas now spawn an effort to go find the protein domain that will do that job. And now you create a new tool.
Yep.
And same thing's happening on the CAR side, something that binds to the target that you care about. On the extracellular side of the cell, pass that signal through the cell, and on the intracellular side, tell the cell to do something. Yeah, in the common case, it's to kill the CAR or kill the cancer cell.
Yeah, one thing I always thought about is, like, for small molecules, we can screen maybe 1 billion different variants. Antibodies, maybe millions. But for like something like a chimeric antigen receptor, hundreds if we're lucky at the time. Or CRISPR, maybe a little bit more than that. And so Serotiny is kind of helping bring multi-domain proteins to parity to other modalities. Because if you can do that, you create different variants, you can test them more easily, and you can then have you can potentially discover new medicines at a faster rate. And that's kind of the key, kind of, at least for me, what got me excited about Serotiny and what you were doing, kind of accelerating the development of these kind of new drug modalities by building a kind of a more generalizable platform and language. And so you have this SaaS product, you can design these proteins, can annotate them, and create new variants, and then you make a pivot towards drug development. Because all in all, building SaaS in biotech is too hard sometimes, most of the time, right? It's like you have to be the exception. And so what was the logic to then say, okay, screw just building software. We need to make our own drugs. We need to actually generate our own data internally. What was the kind of the moment that made you, or maybe set of moments that made you make that pivot? And then we can talk about then kind of how that set up your kind of current trajectory and current kind of updates on the underlying platform and business.
Yeah. You learn a lot by creating a company and jumping into the fire and trying to keep it alive and to make it survive. And so you end up learning truths after a while, where after a few months or a few years, you can distill it down to a sentence, but it takes years to learn that sentence. The therapeutic industry, or the biotech industry, is a strange industry, because you have very few people there are very few purchases or transactions, unless you're literally selling the drug in a hospital. Many biotechs sell a single time to a bigger company or to as an IPO, and so the economics in the market looks different than it does for somebody like Adobe or even Uber or these other companies. So selling software that is especially very specific software, the total number of customers that you have access to is small. And then you have to think about what those customers are selling. And it turns out a lot of times if they're selling a single therapeutic asset once or twice, then if your software doesn't push that binary sale to be more successful or less successful, it doesn't round very well. So it became clear that the valuable thing, if you can move up the value chain towards the actual IP, towards the actual composition of matter, the actual protein itself, you can collect a lot more value. So we in order to get there, where we would start to be building our own designs, we needed to get more data. We needed a wet lab. We needed to demonstrate that what we were doing actually got there. And that in itself is a challenge too, where it's what's so clear to you may not be clear to others, and you have to go and collect the capital in order to collect the data in order to prove what you think is pretty straightforward. And we had a really good team we raised venture capital to do that, a really good team of investors led by Nanodimension, and a really good team of employees to really get that first data to show that we could not only design these proteins in high throughput in the software sense, but that we could actually build them, manufacture them, actually get them into a laboratory and start to test these combinations of tens of thousands of these different designs simultaneously.
Cool. And I think at the time when you kind of made that pivot, I think 2 of the CAR T programs got approved by FDA.
That helps a lot. It's an enormous financial demonstration that what could be built is valuable. So yes, it's not a coincidence that the approval of the first CAR T therapies actually enabled that kind of forward-looking success.
Yeah, I think a lot of Serotiny stories kind of tied into kind of trust between you two and like kind of really expertise, but also timing, being a little lucky. Some other events happen, and then that kind of validates your technology and your approach, and kind of I think it's a lot of startups just kind of lucky events and they kind of taking advantage of those events and riding the wave. And so you pivot to drug development. What were kind of how did you think about kind of building out a wet lab? How did you think about key metrics? How did you think about kind of what types of data sets you need to generate to build better models? That's always a tough problem when you start kind of these, I don't know what to call them, comp bio drug companies, AI drug companies, whatever you call them, but you have to ultimately generate your own data. Yeah. What kind of frameworks? How did you think about for Serotiny? What types of data you need? What type of data sets you need to generate? And how do you scale?
Yeah, I mean, it's you have multiple pressures and multiple problems you're trying to solve for simultaneously with never enough capital to solve for all of them. So in that regard, we tried to build data sets where every single time we did an experiment, we tried to make sure that we were pushing our platform forward. We were bringing data into it, that we would learn something that we could use later, as well as something that actually could be a shot on goal, something that actually you wanted to make sure that there was always a chance. And sometimes it's lower and you're more on one side than the other, but there was always a chance that what you were building or what was in your library was a new useful molecule or new useful protein. So the kinds of things we would do, we knew it was a kind of a blue ocean to go looking for these designs, since much of synthetic biology at that point was kind of done one at a time. That the serendipity that you're both harvesting for the company itself, you're trying to like find those rare events where you get the timing right, but you also can do that scientifically. So we would try to be unbiased about what we look for, and we would go mine inside the human proteome for new components that we thought would be interesting, even when there was very little known about them. There was little literature. And then you try to quickly take them forward. You try to quickly take your hits, and you try to quickly make sure that what you're doing is therapeutically relevant. And so we spent a lot of time thinking about the assays that we were doing. So it was not just the cool design and engineering, but how do you show that the engineering that you're doing actually can help somebody? And so we worked with a lot of people, a lot of mentors, to make sure that the tests that we were running were something people would care about.
Cool. I think shout out to Brian Naughton. We did a podcast of him a long time ago. Shout out to Brian for helping Rodney. I think Brian was pretty important. Brian was really useful for that.
Brian's a good guy.
But you're talking about two things. You said some of the most insightful things you said. You said two things insightful to me. On the first part too, about making that pivot to drug development, I remember you told me, drug development's like an economic black hole for biologists, right? Where it's like if you're doing something cool in biology, you're kind of getting sucked into making a drug somehow, right? It just is what it is. And I think on the platform side, kind of talking about how each experiment enables the next one. It's something really, a couple of years ago, I remember you told me kind of this idea of versioning proteins.
Yeah.
And that's such a powerful framing. Just as much as you version a pair of shoes or iPhones, you can version proteins. And for Serotiny, how do you think about going from a version 1 of, say, a car construct to get into version 10? How do you think about getting there quickly? And how do you think about going from generating a first construct and then thinking about, okay, how do we generate an even better one? How do you even get there? What kind of logic do you use? Is it more kind of interpretation of data, or do you implement some sort of systematic strategy to version things quickly?
Yeah. So at Serotiny, we do both. We go from 0 to 1 and do the invention, and then we go from 1 to and then increment the versions. It was a fun thing to watch. So again, David Liu is the professor who's invented the base editor and the prime editor, and those, by the time he published his papers, already had their version base editor 3 and 4, etc. And we watched that and noticed that it was obvious. It was clear that he was taking code and he was improving it. He would improve the enzymatic efficiency. He'd improve the way the proteins were fused together. The homolog or ortholog of a particular domain that was better suited for what he was trying to do. But the idea of the core had remained the same. When it changed, it got a new name. So we wanted to do that. So in our context, you actually change the assays a little bit if you're going from 0 to 1. You're looking for, does it do it? And so that's much more of a screen. Does it turn on at all? So you can throw an enormous variation into a pool, and if you've created your assay correctly, all you're looking for is any signal at all. Just does it turn on? That's pretty fun. When you get a hit on that, it's pretty exciting. But always you then need to improve it, and that's where part of our platform that's nice is it's not just a screen. We actually make sure that we know how each design behaves, so we can say, let's make it better. So we can put in controls, clinical controls, or our own controls. We can make an enormous amount of variation around the island that you've already found. And you can start to, if you find these islands of stability in protein sequence space, then you need to, once you get that hit, you then need to circle around that island to find the spot that is most efficient.
On the invention, it's just measuring, does it work as we want it or not? Does it lead to some sort of signaling or killing, or does it cut DNA or edit DNA in a certain way, then go from 0 to infinity or something? I don't know. Or 0 to, or go from 1 to infinity. Go from version 1, the best CAR T you can invent. How do you to do kind of machine guided evolution, as Serotiny does? Is that more an ability to do more experiments cheaply, or is that a function of the number of metrics you capture, or maybe a little bit of both? How do you actually, what are the enabling factors to go from version 1 to version whatever?
Yeah, so to play on the term not too strongly, but the idea is you have variation. You try to create that variation, and then you have some selection pressure. You have some assay where you read something out. In general, the proteins we're working with are big, and the cycle time can be long. These are mammalian cells, and can't shortcut a lot of that. So we would always love to improve our cycle time, and we're doing that right now. But our variation is enormous relative to other ways of building these kinds of proteins. So in many cases, the multi-domain protein itself is a very deliberate effort by usually one or a very small number of really smart people really trying to do their best to predict what the best set of combinations of these components will be. But there are an enormous number of combinations of these components. And so what we've tried to do is create you can throw in any idea that you might have a chance of working, but we actually let the system choose which of those many medium to low % working hypotheses will actually surface.
Absolutely. I think the major breakthrough for Serotini's platform versus maybe the state of the art is like, state of the art CAR testing is maybe hundreds of constructs, maybe thousands. And in Serotiny, you can go from millions of designs in silico and then test tens of thousands in vitro. And then that will then lead to, I don't know how many designs, hundreds or tens of designs, but then ultimately you have data where you can go from millions of designs in silico to a functional molecule with, better properties of maybe killing or, efficacy, whatever, engagement, target engagement. And so when you think about creating that variation, what enables that? Is it just is it the software? Is it cheaper? Is it just kind of the way you design libraries? How do you think about maximizing variation? That seems to be the most important metric for that second step of versioning your protein.
That's right. So variation is important. One place we seek variation is in existing protein designs from existing proteomes. If we're going to make a drug, though, we often try to stick with a human proteome, so you don't cause problems. But the human proteome is pretty big, and most of synthetic biology has not explored that. And then that's coupled, looking for variation amongst the human proteome, is coupled with the ability to just synthesize the DNA for these things. And when I was in grad school, synthesizing DNA was extremely expensive. You could do it, though, and you really could. If you had a design you thought would work, you could spend a couple $1,000s to make the design. But with new cloning techniques and with the reduced cost of DNA synthesis, you can start to be clever to actually produce thousands of designs or tens of thousands of designs by recombining some of the sequences of DNA, even if they're very long, not just mutations, but including complete orthologs to each other. And that allows you to build variation very quickly. So there are just over 1,000 type 1 transmembrane domain proteins, or type 1 transmembrane receptors in the human proteome. And these are just your regular old protein that has something on the outside of the cell, crosses through the membrane, and it's got something on the inside of the cell. So it allows some signal to pass into the cell. And there are 1,000 of them. You can start to synthesize most of those, either in components and reuse those components. Can't do it one at a time, but if you can do it in high throughput, you now have access to that whole proteome's worth of tools from a couple billion years of evolution.
Cool. Yeah, I think shout out to Twist for a lot of that work, and a shout out to is Gibson Assembly still used? I think so. Yeah. Is that
use the best tool for the best job? So there's lots of good cloning techniques that help us. Twist has been a useful partner in helping us build some of these libraries.
Absolutely. And so I think it seems like the software enables you to create all these different variants, and then these new kind of new synthesis tools, cloning tools, allow you to test them out, and kind of merging software with kind of new tools allows you to create kind of a whole new platform that maybe wasn't possible 5 years ago or 10 years ago. And so can you talk a little bit more about like you have this workflow to go from maybe an idea, a problem set, to then IP or a product. Maybe we can talk about the business model now. Maybe one thing you could touch upon, let's talk about business model, and we'll talk about data afterwards. But how do you think about Serotini's business model? Because you have this kind of, you start as a software company, go to drug development, and you build out this platform that is really good at versioning proteins, inventing and versioning, do variation of a CAR or CRISPR. How did you and Colin think about business model? Because you definitely are doing something a little different, and be excited to talk about some of the recent deals you've announced.
Yeah. So there's one last step there. So the last step is you have to use the platform in a therapeutic or in a financially useful way. And so initially, we built out some proof of concepts where we went around and tried to figure out what was the best way, the best assays, the best goal for a therapeutic cell therapy. But it was also useful to us to partner with strategic partnerships, people who could provide us synergies where they already have some component that they have characterized and they really understand, or they have some technology that allows them to already understand that goal, the therapeutic goal. So to be clear, Serotiny was started as more of a synthetic biology company. That's been our expertise at building the platform. And we've actually brought on some great people who have that therapeutic expertise at this point. But in the beginning, that was where we thought we could partner with those who already have that therapeutic clarity. And the two places where this has been obviously useful are, again, the gene therapy context or the cell therapy context. With the gene therapies, one of the big goals is to be able to insert sequences into DNA with an arbitrary payload, make them how you want. And so we've been working with Tessera Therapeutics, a really powerful flagship company who has found some really interesting ways to get these gene therapies up and running. And so we're going to work with them to on their multi-domain proteins for gene insertion. And we've also, on a different side of things, worked with have a relationship with Janssen Therapeutics, Johnson and Johnson's R&D side of things, where we're going to help them build cell therapies with improved and novel receptors for these cells. So, and in both cases, we're able to talk about that process that you talked about, where you start with kind of a conception idea of what you want to achieve. You build out an enormous library of variations to test as many different hypotheses as you can all at once. And at the end, you're left with something that is a therapeutically useful multi-domain protein.
Cool. Yeah, well, congrats on the partnerships. I'll put the links into the description of the deal announcements, and I'm sure there's a lot more. I know there's a lot more in the pipeline left. And maybe you have these partnership. That's one part of the business model.
Mm-hmm.
You use a platform and you go to a partner and say, okay, they want to do something in a specific problem set, whether it's in cancer or maybe a specific modality. Or how do you then think about sequencing those deals and stacking those deals? That's kind of the art of biopharma deal making. Maybe Colin spends more of his time on that, but kind of the art of the deal. And then how do you think about external versus internal versus something we'll do partnering with versus something we keep internal to Serotiny?
So there are a few features to that. So one is, in our case, what we have is we're starting with a platform, and we'll acknowledge that straight up. This is a platform technology. And I think there's always this push towards, reversion to the mean, where platform companies want to make therapeutics and therapeutic companies want to have the ability to make more than one by showing that they have some kind of platform. And so when does a therapeutic company build out its own pipeline versus when does the therapeutic company branch backwards? So that's one feature. The second is the state of the industry. So if you build out a platform company, you need clients, you need partners, you need customers. And so it's been fun to watch the industry see the value in the potential of these cell and gene therapies. And we've watched that happen before our very eyes, and that has enabled these valuable partnerships that are more than something like a software as a service from the early days. It's clear that sometimes the product itself can, in the therapeutic context, can be valuable. And I think that our platform is close enough to the therapeutic material that we can start to push in that direction. And we've brought on the expertise to help us do that. And we're looking to build these synthetic biology tools the control cell behavior in a way that improves these therapies beyond what currently often were built as one-offs, and they're sufficient, but we think we can really improve upon them. And the space is big. There's a lot of things that a programmed cell or a programmed protein can do. A lot of different targets, a lot of indications, and whole new sets of modalities that are enabled by this kind of programmed biology.
Yep, absolutely. I think partnerships help scale platforms in a less dilutive way. Dilution, and then is really important. My worst enemy, my worst enemy. I have nightmares about dilution, and nightmares. I have a cold. I wake up in cold sweats about recaps and stuff like that. But then on the opposite side too, once you get scale, you can then do experiments that no one else can do.
Yeah.
And it's some of the data for Serotiny. It's exciting. Kind of shown an ability to invent a new construct, version it, and then generate some really exciting data internally. And so I think the world will begin to see all the stuff you've been doing the last few years and begin to appreciate, well, once you get scale, you can start doing things that maybe you could screen a larger number of variants, and that can lead them to kind of constructs that other people just couldn't even discover at all. And enable new types of cell therapies and a lot more.
There's back to the kind of timing issue. It's nice to work with technologies or to ground our own work on technologies that are themselves scaling. It's a little scary because you don't want to be caught where you need that technology, but it hasn't scaled yet. Yeah. But it's a really nice feeling where you can ride the wave of the technology itself. So software helps us. That's already scaled, but everybody knows how to scale that. But it is scalable, so we can scale our software pretty well. The synthetic biology side, again, is scalable, and it's taken a while for it to become as cheap as it is to synthesize the DNA. And the sequencing itself also is scalable. You can crank that up, you can parallelize it, you can do many projects all at once. And so that is another feature of kind of the platform that allows you to take on lots of different projects and not just have all your eggs in one basket.
I think it's a really nice place to be a market leader in a emerging category. I think Serotiny is the market leader for the design of multi-living proteins, right? Just the data shows that, and you have the technology tools to do that. And so, I think as cell therapies get more successful, Serotiny you have a lot more success. And so, okay, we talk about Serotiny, talk about your kind of the platform, the business model, kind of any outstanding problems, as you kind of built this company up, but that just more generalizable to other companies. One thing I see in all these companies is this idea of like how to actually generate the right data for models. Buddy Jacob Oppenheim from ECRX, he had this concept of doing the unphysical experiment to set the boundaries for models. Do the experiment that you wouldn't really do, that doesn't create value, but it's valuable for a model to kind of know the boundaries. Like any problems like that, maybe that's a problem, but any more generalizable problems you're kind of experiencing at Serotiny that like maybe other companies are facing, and kind of any ideas to solve them, or maybe startup ideas and stuff like that.
Yeah, I mean, I think that it is important to be able to communicate your results to others as much as it is to be able to generate really cool science. And there's always a little bit of a tension between doing an experiment that's obvious, that's easy, that will be less incremental, versus doing kind of the right experiment, the big way, that's going to be expensive and require a lot of resources. And I don't actually know that there's a right answer to when you have to choose. Do I go all in on the one experiment that's going to be the big one, or do I do a few smaller ones with the budget that I have right now? But it's a choice that you constantly have to weigh. And you have to make those choices not just based on the good science, but on considerations of communication. You have to be able to communicate to your employees and to your investors and to your partners. And so there are really good examples of creating data that helps you get a deal, or helps you get fundraising, or helps you hire even, that allow people to see inside of what you're trying to do.
Interesting.
And so I think it's one of the lessons there.
That's pretty insightful, actually. Actually,
Thank Colin for that one. That's Colin and I spent a lot of time chatting about a lot of these ideas. So
even
you've
me,
got two brains here.
You have this idealized thing of platform
one.
companies that you're supposed to generate these huge data sets and and be comprehensive. But then when you're in the weeds of building a company, sometimes you need to generate the data that is valuable at the time. And kind of balancing the act between doing something that's systematic versus kind of what's kind of needed. Any types of anecdotes or stories from Serotiny you can like discuss that like maybe are like that? Nothing confidential though. If it's all confidential, we can just go beyond it. Just talk about something else.
I think it's been fun to a little related, but not entirely related. When you get to be unbiased and when you get to kind of let a platform that you have shine, and you trust it, and you have to trust it because you don't know what at the end of the day the biology wins, the science wins, reality wins. You don't get to tell it what to do. But when you say, look, I think there should be something here, and you run the experiment and you pull something out, it's often pretty exciting that you get a hit or you get a result that matches your theory. And what's really fun is when you go back and you try to figure out, what's going on here. You realize that there was ambiguity in the literature, or the literature would never have predicted this, that this would have been way down on the list of what would have been expected. But that by trusting the process, by doing the right experiment, by letting the science work itself out, you found something in a way that other people couldn't, and you found something that's impressive. So it's a good feeling when you do get to run that big experiment that really proves your point.
Cool. Okay. Great answer. And one thing too, like you and Colin, you guys progressing as leaders, where you guys, I can see you guys as public CEOs one day. You guys have good answers. Like you're getting the public CEO persona. I love it.
All right.
Can I ask you a question?
I don't know if I'm working on that, but
example, and you just gave me some cool kind of overview. I love it. Maybe talk about more about your transition as leaders. Last thing to talk about. We talk about one thing I love about you two. You guys such empathetic leaders, incredible managers. I've seen you kind of really be thoughtful about team building and who to hire, how to construct culture. What have you learned as going from a grad student to build Serotiny as a SaaS company, to all the way to leading this huge team, recruiting executives? Any kind of lessons you learned from becoming a better leader? Because I'm definitely seeing it, right? You and Colin have become definitely better leaders, and you guys are going to get better. Any kind of key lessons you learned journey so far?
Yeah, I mean, it's a different job. So as a scientist, you're trained to do science, and that's one thing. When you jump into the deep end, you have to learn these other things. You have to want to do these other things, and you don't even know how valuable they are until you get into them. The business side of things is really important. BD is a whole different language. Leadership is a whole different skill set. And I think it's at least from my perspective, it's been helpful to be humble. It's been helpful to acknowledge that I'm new at this, and I'm learning too, and actually have people. We've had great employees and friends and mentors who have coached me and said, hey, we can't be doing that. We got to be going this way. It's like, I see, and I appreciate that. And that helps a lot. It's a weird transition, a very strange transition, to go from being the person writing the code to helping other people who are much better at that than you, give them the autonomy and the authority to run with that. And it's really fun when you get to see a whole team working, and you get to see so much productivity that you could never do yourself, because you've been able to bring such interesting people together. So I think being humble is really important. I think learning from your mistakes and learning from your lessons and acknowledging that you'll make those mistakes is really important. And making sure everybody is on the same page and has the right incentives is very helpful. And we'll keep learning. I don't think I'm done with my lessons yet. But
I think one thing too, people just like being around you. I think it's probably something that's underrated too. think people just like being around Justin. That's what
All
I'll say.
right.
I think a lot of people just like
All
hanging
right.
out with you, working with you, just pleasant person. And I think one big moment I observed, a big definitive milestone in terms of that leadership transition was when you and Colin got new headshots. Okay, got some new headshots. Looking good,
Yeah.
guys. I
The
got new
publicity
headshots.
part, we're
I
also
got new
learning about
headshots,
too.
right?
So.
It was a big transition in my career when I got new headshots. I think it's a big moment
Mhm.
for anybody's career is when you finally get a professional headshot done. It's like, okay, now you're a whole different trajectory, whole different era.
You don't want to do it too early, but you also don't want to do it too late. So,
Well, yeah, one
it is
recommendation to
important.
everyone: get professional headshots as early as possible. Yeah, it's something I didn't even know. I just took a picture. Then go spend the $500, $1,000 to get a good headshot done. It's invaluable. So one leadership lesson too. And then could you talk about maybe like last thing in terms of like hiring?
Mhm.
The part of this leadership part. One thing I'm observing is the biggest bottleneck for startups is no longer money; it's just team building, hiring. What kind of some tactics you've used early on to hire? You recruited a I forgot the CSO's name from Kite or no
Jeff May from Maverick.
from Maverick. Yeah, so you've recruited some really top-notch executives. What were kind of some tricks and lessons you've learned from hiring, which is just so hard for every biotech company, no matter how much you've raised, whether you're seed or you're a big mega company, like hiring is so hard.
Yeah. There's a lot of interest in talent right now, and there's a lot of people, especially on the science side, who may not realize quite how valuable they are to the industry right now. There's just such a need for people with skills to do things like mammalian synthetic biology that traditionally weren't a job that would pay very well. Uh, I think it's important to know what you can offer as a company and be clear about it. And there are some things that we cannot offer as a company, as a small to little startup, and be clear about that. And if you're clear about it and other people know what they want, you can arrive at a place where everybody, I think, can get what they want. But if you're not clear about it, or if you try to compete against the big companies with resources that you don't actually have, I think you can run yourself into trouble.
Yeah.
So we try to be very clear. I think tapping into social networks as much as possible, even though that can be hard, is really helpful. Your friends and recruitment and friends of friends is often very helpful. Before COVID, giving talks and I guess that's coming back now, which is great. Seeing people in person, meetups and things like that can actually be really a nice way to meet new people and get out of your normal comfort zone.
Totally.
It's a hard thing to do. Very important though.
One thing I'll say, what I love working with you, Colin, and I think a lot of other people do, is your high integrity. So people know with the Farlow brothers, high integrity. It's like you trust them. They're gonna tell you, going to be transparent about what they're doing. I think that's the reason why people are attracted to Serotiny. It's just like it's a very high integrity organization. I think you've built that in your culture. And I think this podcast and a lot more in the future is you're already a superstar. Now it's just about unveiling that. There's no very easy work. So I think in the next few years, you're going to be on some more panels and jet setting to, I don't know, you'll be at JP Morgan. We'll go JP Morgan together.
Okay.
You'll be on some panel. A lot of panels. We'll be on famous co-founder, and you'll be on CNBC or I don't know, TV shows and stuff. Bloomberg or whatever. I always want to be on Bloomberg, actually. Let's do Bloomberg one day.
All right. Well, I appreciate your optimism, Josh.
I'm so optimistic. You know how optimistic I am. I'm so positive.
It's great. No, it's wonderful. It's a good quality.
I'm your movie agent, so I gotta be hyping you up. So okay, we're gonna publish this. This is a Friday. I'll send this on Monday, and we'll get the hype cycle started. We'll do this again. I appreciate you doing this. I'm gonna press stop record right now, but and we'll talk.
Well, thank you very much, Josh. Really enjoyed chatting with you. I think helping the community understand how these companies are built is helpful. I think there are a lot of people who are interested in it. I think the community is still pretty small, and I think you've done a great job of trying to get that communication. So I appreciate it.
Catalog of stories, and this is gonna be useful like 10 years from now. Like they're gonna be like, whoa, we're gonna unearth. It's gonna and
see how much we were wrong. How much.
No, there can be so much.
We didn't know what we were doing.
You know, take the Disney Vault. They're gonna be so inspired by Justin Farlow's story, and we'll do a follow up. And you know, this podcast will last 1,000 years. Aliens will come down and listen to it. I'm like, wow,
I'm scared about that, but I do appreciate. I would like the ability to come back in a little bit and see what else we've learned. So.
We're gonna do it. Okay, I really appreciate it. Let me press stop record right now. But I appreciate this.